Good evening, everybody. My name is Sharik Haider, and I'm the president of CFID, and it's a real pleasure to welcome you this evening to our COVID-19 vaccine webinar. Just want to make sure that you can see my slides here. Give me a second here. Apologies. Are we able to see my slides?
Yes, we can.
Okay.
They just need to be in presentation mode.
Right, there we go. All right, well, again, I'll start that again. Good evening. My name is Sharik Haider, and I'm the president of CFID. And again, real pleasure to have you this evening to join us on this COVID-19 vaccine webinar. This evening's webinar is co-sponsored by Moderna Pharmaceuticals and NCCID, and the National Collaborating Center for Infectious Diseases. But the content of today's vaccine webinar was developed entirely by CFID and its board members. I want to tell you a little bit about CFID before we get into the actual webinar. So CFID is a registered charitable organization. We were first established in 1989. CFID is committed to improving the health of Canadians and certainly people worldwide through the support of research and dissemination of knowledge pertaining to infectious diseases. Now, we have a number of major activities. Certainly the most important one is fundraising to support our mission and goals, certainly promoting knowledge translation and dissemination. We fund innovative research, and we'll speak a little bit about that shortly. And we certainly like to educate through lectures, seminars, and webinars like we're doing this evening. One of the things that we certainly take dear to our mission is certainly our role in mentoring and supporting students and trainees, who for sure are our future as we move forward in this field. Now, CFID has a number of activities that we support. So we support a number of grants. One specific one is the Safe Water Drinking Grant that takes place every 2 years. We certainly support other theme-based grants as well. We do award trainees opportunities for research over the summers. This includes undergraduate students, medical students, and most recently we have awards for internal medicine residents and pediatric residents to attend their national conferences. We have as well faculty mentorship awards. Like tonight, we do a number of webinars in terms of helping disseminate knowledge and experience, and we host workshops, and some of the workshops that we've held have been very well received. They include workshops for faculty, for new faculty, and for those that are mid-career. I think our signature CFID activity that many of you are familiar with, though, is our CFID bug run, and this has been around since 2015. Now, during the COVID period, we actually moved to a virtual CFID bug run, and many of you were kind enough to send us your pictures in your virtual runs that you did in your homes and your favorite places, and you can see some of these pictures that are up here. In this past year, we actually managed to have our national meeting in Vancouver, and we held a hybrid CFID bug run, so you could either do it in your own home, and there was, in fact, a run led by our past president and certainly dedicated runner, Dr. Curtis Cooper, and that went exceptionally well. I'm not going to go through our CFID board of directors, but I wanted to recognize them. This work could not be done without the help of a number of very talented individuals, some of them that are here this evening co-moderating with us. And I think it goes without question in saying that we could not put on events like we're putting on tonight were it not for our partners and supporters. I've already spoken about Moderna Pharmaceuticals and NCCID supporting tonight's events, but as you can see here, we have a number of important partners and supporters, and we're truly grateful for their support of our mission statements as we move forward. So with that in mind, I would now like to move forward with some introductions. So I've already spoken about myself, but I have the pleasure this evening of co-moderating this webinar with two other speakers, Dr. Curtis Cooper, who's from the University of Ottawa. He's an infectious disease specialist, professor of medicine, and past president of CFID, and Dr. Marissa Becker, who is from the University of Manitoba, also a current board member of CFID and an associate professor of medicine. It gives me great privilege to introduce our three amazing speakers for this evening. I think you're in for a real treat this evening. Lots of very valuable information to be shared. Our first speaker is from the University of Ottawa, Dr. Mickey McGinty, and she'll be speaking about an overview of the landscape of COVID vaccines, certainly what we have present today and what's coming down the pipelines. Our second speaker, I know a topic that's I'm sure of interest to many of you, is the mRNA vaccine platforms. And we'll get into discussions around distribution, access, and broad applications. And our speaker this evening will be Dr. Jason Kindrachuk, who is the Canada Research Chair in the Molecular Pathogenesis of Emerging Viral Infections from the University of Manitoba. And it gives me great pleasure to introduce our final speaker, who may not be visible to you this evening. She will be joining us very shortly. It's a colleague of mine from McMaster University, Dr. Susan Wasserman. She's actually the division director of the Department of Allergy and Immunology. And she'll be speaking about a very practical topic that I know all of us in infectious diseases are facing on a regular basis, is the topic of adverse reactions to COVID vaccines, how to identify them, how to deal with them, and then that question that comes up about exclusions from COVID-19 vaccines. So that will serve as a nice overview of this evening's webinar. And so without any further delay, it gives me great pleasure now to turn it over to our first speaker, who will be Dr. Mickey McGinty. Look forward to your talk, Mickey.
Thanks very much, Dr. Hayter. You've got to stop sharing so I can. Here we go. So I'm going to take us on a whirlwind tour through COVID vaccines and all of the data that has come out over the last 2 years. So this is going to be a very high level overview of vaccines for COVID-19. So if there are questions generated in the Q&A, hopefully we can come back to some of that, but hold on to your hats because I'm going to talk fast. The only disclosures I have to mention is that I do sometimes take speaking fees for other kinds of speaking engagements, including from Merck, and we did do a vaccine clinical trial at the Ottawa Hospital of a COVID-19 vaccine with BBI vaccines. We're going to talk about what's available right now in Canada, how they're administered, as well as we'll talk a bit about immunogenicity and not so much about reactogenicity because Dr. Wasserman will touch a lot more on that later on. We'll look at how efficacy has evolved over the pandemic, as well as durability as it gets into what's the future of using these vaccines for management of an ongoing persistent or recurrent respiratory illness. And some peek behind the curtain of what's coming next or what the strategies for dealing with the need for ongoing immunoprotection against COVID-19 will be. So there are at the moment 6 vaccines that you can obtain in Canada. There are 2 mRNA platform vaccines, which Pfizer and Moderna. Each had a 2-dose primary series, and as you are aware, most of them, for most of these, booster is recommended of at least 1 additional dose, but in some cases and in some populations, we would consider adding 2 additional doses of an mRNA vaccine after receipt of one of these others. So AstraZeneca was originally a 2-dose series. In Canada, most patients got 1 dose of AstraZeneca, and that was followed up with a 2nd dose from an mRNA series. Janssen is the only one in this list that's a single-dose vaccine. You can see its efficacy in the pre-Omicron era was a bit weaker than some of the mRNA vaccines, but it's a different kind of viral vector vaccine and didn't come with some of the baggage that AstraZeneca's vaccine vector had. Medicago is the newest. It's a plant-based virus-like particle that was developed in Canada. It's a 2-dose primary series as well. And then finally, we have Novavax, which is the more traditional, more commonly familiar type of vaccine called a recombinant protein subunit vaccine, which is here to save us. And maybe Dr. Russman will talk about it in the context of patients who cannot get mRNA vaccines. There's some alternative therapies out there now. So the traditional way that we decide how well vaccines are working is to look at how well we generate antibody against the pre-specified target. So the first step in that is, do these vaccines generate high levels of antibody titer? And the answer, of course, is yes, they do. So this is a graph from a more recent summary article about immunogenicity with pooled data from the original clinical trials of these vaccines, showing that the mRNA vaccines generate lots of high-level specific immunity, but the adenovirus vaccines are no slouches. They develop pretty good antibody levels against spike protein, and that those antibody levels correlate quite well with neutralizing antibody titers, which really just gives us the confidence to use the easier to measure antibody level as the outcome measure or surrogate outcome measure as we need to. All of these vaccines separately have been demonstrated to stimulate T-cell immunity as well. So we get good CD4 T-cell recognition of the antigen after all of the primary series of these vaccines. That's important because we know that durable immunity is not just a response from your immediate humoral response to vaccination, but it requires ongoing coordinated effort from your cell-mediated compartment. It also helps us to know that patients who can't generate humoral immunity may still benefit from vaccinations, although the efficacy might be affected. And we know that this is true because even in patients getting anti-CD20s, when we vaccinate them, we can measure these responses very detectively. Reactogenicity in general. So I think in my mind, it was always, how does this compare to other vaccines we ask patients to take? And the reality is there's a lot more reactogenicity, a lot more local and generalized systemic side effects. And by more, I mean more frequent. mild side effects after first and second doses and even third doses of these vaccines than you would expect to see from the other vaccines that we commonly use in the marketplace. So you can see flu and pneumococcus are shown here. And the only one that's worse than the average COVID vaccine is the shingles vaccine, which if any of you have had it, may be able to attest to that. I wanted to touch on some of the hangups we have about our current vaccine strategies for optimizing immune responses after COVID vaccination. So as you may or may not know, after we are vaccinated and our humoral immune compartment starts to generate antibody, there's a quite complex process that happens between your T cell compartment and your humoral immune compartment, which helps refine the quality of the antibody and the lasting memory affinity of that compartment. So it's not new to understand how that relationship works. We've been vaccinating people for a very long time. And if you reflect on typical vaccine series that we give to adults and children, you'll note that often we leave longer intervals between vaccines and boosters than we have with the COVID-19 vaccine. And in fact, it's quite easy to demonstrate that with these vaccines as well, the longer the interval, the better affinity maturation occurs and the more durable that immune response might be. So here's some real world data to look at with respect to that from BC's registry, which shows that patients who got mRNA vaccine with Pfizer had vaccine efficacy that varied with the interval between the two doses of the vaccine in the primary series. So at 3 to 4 weeks, which is what was typically being recommended in the middle of the pandemic, because that's what was studied in the clinical trials, the vaccine efficacy is markedly reduced compared to waiting longer. So after 2 months, you probably maximize the amount you need to wait for good vaccine efficacy. But we continue to vaccinate people on a Q0 and 1 month schedule for their primary series. As far as reactogenicity, this study is trying to convince you that you also see a reduction in the reactogenicity if you wait longer between the doses of vaccine, which I might buy based on where some of the dots are falling on this line, but it's a hard sell that that's a big advantage to waiting on the vaccine. I wanted to bring up another special population because it's of an important interest. So immunocompromised patients, in particular solid organ transplant patients and patients with renal failure, have demonstrably and markedly lower antibody responses to primary series of vaccines. So what you can see from this study out of the New England Journal, looking at solid organ transplant recipients, that is after their first dose and right before their second dose of vaccine, there's not very much detectable antibody. Whereas before the third dose of their vaccine, you get quite a bit better. And after a month after their third dose, they get titers that are quite comparable to what immunocompetent patients will generally experience after their first 2 doses of vaccine. So this is the data that was used to suggest that these patients really need a 3-dose primary series and then may need additional boosters after that, as they tend to have waning that occurs more quickly than other populations do. We also have had the opportunity over the pandemic, partly because of available supplies, but partly because of scientific interest, to look at the difference between vaccine efficacy, but also immunogenicity after mixed regimens of vaccinations. So this is just, but this particular example is looking at what happens if you mix infection and vaccination. So in this study, what they looked at was people who had been infected with COVID-19 before receiving a first dose of vaccination versus patients who had not been exposed to COVID-19 beforehand, so who were seronegative for antibody before the first dose of vaccine. And you can see that adding a vaccine after a patient has had an infection induces a bit of a higher antibody response following the first dose of vaccine, which is logical in the sense that their immune system has already been primed with a first dose of vaccine or a first dose of antigen when they got their infection. But we can see the same sort of thing occurs when we mix vaccine types as well, in particular when we mix the ChAdOx or the AstraZeneca vaccine with an mRNA vaccine as a booster, we do see higher levels of antibody generation after vaccination with the second dose. But how long do these antibodies last, and how long can they protect us for is a more important question. So it's important to note that protection against hospitalizations and severe disease is much more durable than protection against symptomatic infection, which may be truly an effect of durability and a wane of antibody titer in response to COVID, but also it can't really be untangled from the effect of exposure to new variant strains, which may have some immune evasion of the vaccine antibody that we have that's directed against wild type SARS-CoV-2. This is a nice study that looked at modeling of antibody decay rates following observed data in primary vaccine series. And from this study, and vaccine effectiveness and infection rate outcomes, they were able to say that about 20% of your convalescent titer, so the 20% of the titer that you see about 28 days after the second dose of vaccine, is needed for protection against symptomatic disease. But only 3% of that titer is needed to prevent you from going into a hospital because of COVID-19. So I'll pull that up a little bit better with the labels on there for you so you can see it a little more clearly. So if we're looking at the half-life on the time axis, we look at 250 days, which is about where their data ends and their modeling begins. And you can see that the efficacy against mild infection, which are solid lines, versus severe infection, the dotted lines, requires a quite different antibody titer response. We can get into that a little more if we need to. This is just to show you that they're not making that data up. So over the 6 months after your second dose of vaccine, there's a steady decline in antibody titer, accompanied by a steady decline in neutralizing antibody titer over time. T cell responses don't fall. So here again is the antibody response, looking at different combinations of vaccine exposures. But you can see the trend is all the same. No combination of vaccines as a primary series protects you against the decay of antibody over time. However, T cell responses persist and don't have nearly the same decay over the time period, and are equally measurable at the 6 months or longer time point. So T cell responses are the color coding on these lines, so it overlaps a bit, which makes it a little hard to see, especially if you might be colorblind. But the T cell levels for low are pink, for green are intermediate, and blue represents high T cell antibody responses. You can see they track. If you had a low response, it stayed around the same level. If you had a high response, it stayed around the same level over time. I know that this chart is really hard to see overall, and I really just wanted to bring it in so it would prompt me to remind us all again that efficacy against severe disease and symptomatic disease has diverged dramatically over time. And again, this is as likely to be contributed to by the fact that the Omicron variant is not the virus against which these vaccines were designed, as well as the period of time between which people were vaccinated with their primary series and had exposure to the wave of infections in our community that was represented by Omicron. So if you look at vaccine effectiveness, it's all very high in the wild type, in the Delta period, which is the upper sort of 2/3 of this graph. And only the bottom 4 sliders here are showing you the vaccine effectiveness against symptomatic infection against severe or symptomatic infection with Omicron. So if you're immunocompetent or immunocompromised, 2 doses doesn't protect you nearly as well as 3 doses does, and 3 doses gets you pretty close to the same level of protection that we were seeing against Delta and wild type virus with our classic series or classic vaccines. So we can say that that protection is partially restored by giving people a third dose of vaccine. And then the question, of course, becomes it's just more of the same. And I'll skip over it for a time. The question then becomes, of course, if the third dose does so well, how well does the fourth dose help? And I haven't pulled out the studies to show you in much detail other than to show you that we'll talk about what happens with more boosters at the end as we get into future directions. But the challenge now will be in vaccine development going forward is how do we know how much antibody is going to be needed to protect us against COVID-19? And this is a bit of a moving target because it's likely that different variants will have different correlates of protection. But it's also frustrating that the vaccines modeling based on the individual vaccines and their effectiveness leads us to quite dramatically different expectations of what the correlative protection should be. So I'll show you the simplest study that has the clearest study that has looked at this. And you can see that when they try to model or overlap the antibody peak responses against vaccine efficacy, they can model what the mean antibody that appears to be required for protection would be. And in this case, it's about 298 BAU, which is the international standardized units for antibody measurement in this for COVID-19. But as you can see from the dotted lines around that nice green solid line on this graph, the margin of that result is quite wide, and it's probably so wide as to prevent us from having a lot of confidence that a patient with an antibody level of 300 or 298 is actually going to be protected against symptomatic infection. Moreover, all of this data is effectiveness against Delta or wild type and is not, this sort of modeling is not yet available for Omicron. There's a study that came out to try and standardize this, looking at all of the different vaccine models and trying to come up with a correlate that could be used not for clinical protection, but for comparing future vaccine platforms to make sure they met a minimum threshold of immunogenicity, such that we would be able to compare them and have some expectation that their clinical effectiveness would be comparable to the vaccines that we've used so far in the pandemic, mostly because we won't have naive patients to vaccinate anymore. So we're going to have to use surrogate markers to help us figure out if our vaccines are working in a timely fashion. So what comes next? So more boosters. So they did try this strategy in a couple of places, Sweden and Israel notably. In Israel, the data that's published and available so far is very short term, although I'm sure very soon we'll see the longer term data related to fourth doses of vaccine. But you can see that a time after fourth dose did seem to improve protection against severe illness. But symptomatic infection, you got a bit of a bump, but it waned pretty fast after that fourth dose was given. There are lots more vaccines in the pipeline. At the moment, there are 166 COVID vaccines in clinical development and almost 200 in preclinical development. 63 of those vaccines are at least in a phase 2-3 of their research programs. So we're still going to see more to come. But maybe we also need more strategies for vaccinating people successfully. And one of the ones that was quite exciting throughout the first, I would say, year and a half of the pandemic was asking ourselves why there seemed to be a strong correlation between countries where BCG vaccine prevalence was high and lower COVID-19 severe outcomes, because it was well documented up to now that BCG vaccine has a non-specific effect on reducing deaths and illness, morbidity attributable to viral respiratory diseases. So there was a lot of hope that a BCG vaccine might provide a cheap, safe protection mechanism against COVID-19. And in unfortunately, the first randomized clinical trial that tried to answer this question showed us pretty convincingly that BCG vaccination had no effect on people's likelihood of getting a COVID-19 infection. This data is from 2020. So it's from the era where people would not have received other vaccines. Moreover, it's South African data, so it's well before they would have received specific vaccine against COVID-19. And they didn't observe any benefit in terms of COVID-19 infection or hospitalization in patients who got BCG. But now that we're moving on from that one, there's interest in other vaccines that may be providing cross-protective benefit or off-target effects with their vaccine strategies. There was a nice study, which is a retrospective cohort analysis of California patients during the first 6 months of the pandemic, 3 months of the first 6 months of the pandemic, who had received pneumococcal vaccine with either PCV13 or both PCV13 and PPSV23, and looked at and observed that the hazard ratio of COVID-19 infection, hospitalization, and death was lower in patients who had been given PCV13. And all of these patients were high risk because they were all over 65 and had received PCV13 vaccine after the age of 65. So now it'll need its own randomized controlled trial to help us prove or disprove that this is true. But I will say that this is not the first time that this vaccine has been implicated in this response, either in studies of maternal vaccination of influenza. They did observe that maternal vaccine with influenza combined with infant vaccine series for pneumococcus resulted in the lowest number of childhood respiratory infections. So something about off-target effects or non-specific immune activation related to these vaccines may indeed provide protection against off-target viruses. But in summary, there's lots of vaccines we've engineered for wild-type SARS-CoV-2. They provide quite durable protection, even with only primary series, against hospitalization and death. But time and variant evolution do weaken their efficacy, in particular for symptomatic mild illness. More doses might help that, but it seems to be that the added protection after a third dose, so with fourth and subsequent doses, is likely to be shorter lived. That may be a result of antigen fatigue or simply a loss of efficacy because of the wrong target being repeatedly exposed when we're trying to protect ourselves against new variants. But we are going to need to find a strategy for managing the risk of severe illness in the future. And I think we still have yet to define at what point after the receipt of your primary series, the population risk becomes high enough that we need to seriously consider boosting and alternate strategies. Hopefully novel vaccine candidates and platform will bring us a more universal vaccine in the future. I didn't pull it up, but there are about a dozen vaccines that are not for SARS-CoV-2, but are for that are meant to be variant proof or pan-sarbecovirus products, which are mostly in preclinical, but some have moved into clinical development stages of their research. So those may be coming in the next 2 to 5 years as well. And that's it.
Great. Well, thanks a lot, Mickey. That was, as always, a great overview of the COVID vaccine scene. We have time for a few questions. I encourage people in the audience to submit their questions. We'll start off with a question from one of our colleagues here in Ottawa, Dr. Greg Rose. Thanks for joining us tonight, and thanks for all your good work with the OMA on our behalf. So Greg has a really important question. Are there data or recommendations to suggest that previously vaccinated patients undergoing marrow ablative chemotherapy require a repeat primary series?
I would love to tell you that there is detailed observational data that tells us for sure. There is not. I can tell you that anecdotally, I mean, they lose their antibody responses when they go through their marrow ablative procedure. So they will lose protection. How much more benefit do they get from additional vaccines afterward is a question that is not, as far as I'm aware, being asked in a clinical trial format. Most transplant centers simply recommend that that vaccine series be repeated or have a hybrid vaccine series for patients who started their series before their treatment. The challenge is that there aren't enough of these patients, and the virus has even in those populations become so much less likely to result in clinical illness and hospitalization that it's become very difficult to measure those outcomes with meaningful accuracy.
Great, thanks. We have another question from our colleague from Calgary and CFID board member, Dan Gregson. Can you comment on intranasal vaccines that are in development?
Oh, yeah, I didn't have time to tackle all of the cool new strategies. So intranasal vaccines look really promising in preclinical studies. It makes a lot of sense, just like with influenza intranasal vaccination, to use a vaccine that's going to stimulate local immunity and maybe offload some of the risk of reactogenicity that we see with the systemic vaccine platforms we're having to use to get these effects. So I am really looking forward to the phase 3 results that I think we're expecting to see like pretty soon for a couple of those platforms. The other thing it will give us, even if we have a hard time measuring its efficacy against true infection in this brave world where we all already have vaccines, is it will give us new antigens too. So as far as boosting regimens, it may help improve the efficacy of additional doses of vaccine.
Okay, good. Sherrick, do we have time for one more question?
Yes.
All right. So which one to ask? I've been busily writing down ones, and I guess I'll ask the question, why aren't we updating the antigens with the currently available vaccines? We do this for influenza with, seems like sound rationale. Shouldn't we be doing this for the coronaviruses too?
It's a really fantastic question, and I'm glad you asked it because I know that Jason is going to tell us a lot more about that in the next part of the presentation. But with influenza vaccine, we demonstrated that we can turn antigen around pretty quickly and scale it into population level vaccination, at least in North America. A lot of these strategies leave large parts of the world behind, and I didn't really have time to touch on that part, but we definitely can make new antigen-specific mRNA vaccines, and one of the Jason will tell us more, but one of the promises of mRNA technology was supposed to be that it could be scaled relatively quickly. So hopefully Jason will give us some insight about what's coming with variant-specific vaccine.
Great, thanks. So once again, thank you very much for a great talk. And with that, we'll say bye to Mickey, and we'll welcome Dr. Jason Kindrachuk, who will now talk to us in a little bit more depth about mRNA vaccine technology.
Great, thank you so much. I'll just wait until I have permission to share my screen. But I do want to thank Mickey for two things. One, for putting a lot of pressure on me for what I'm about to present, but also for having her cat walk partway through the screen. I'm at home right now with a sick child and single parenting for the evening. So if you see a child or dogs kind of run in behind me, you'll understand why. Listen, I'm Jason Kindrachuk. I'm just a virologist. I dabble in a number of different viruses, but obviously have been pretty involved in COVID-19 now, getting fairly involved into monkeypox as well. So let's talk a little bit about the landscape for mRNA vaccines. One of the things I first want to do is certainly, obviously, bring up the fact that being as part of the University of Manitoba and here in Winnipeg, that I'm located on the original lands of the Anishinaabe, Cree, Oji-Cree, Dakota, and Dene people, as well as the homeland of the Métis Nation. So let's talk about coronaviruses. Coronaviruses, first of all, I think we all know at this point what they are. They belong to the family Coronaviridae. Their RNA viruses have very large genomes. They're positive sense RNA viruses with a single-stranded RNA genome. So that makes them a little bit different from what we see with things like the filoviruses and things like Ebola virus, the influenza viruses, which I normally work with. But where things get a little bit complicated is when we start to try and parse down what the different coronaviruses look like, because they're a very diverse family of viruses. They're currently divided into 4 genera. That's the alpha, beta, gamma, and delta coronaviruses. But when we look at the different genera that are known to be associated with human diseases, so far it's only been the alpha and the beta coronaviruses. But we can appreciate that even amongst the alpha and the beta coronaviruses, these are fairly diverse sets of viruses. Coronaviruses, by and large and part, usually cause respiratory illnesses in humans, as well as gastroenteritis in animals. We have to consider that when we think about coronaviruses and we think about the impacts in humans, we have to also appreciate that these are viruses that have a large impact on livestock. So when we think about the global health impact of coronas, it's not just related to, necessarily at least, to those that are directly causing infections in humans, but we also have to appreciate the effects that we see on animal health, and obviously the reliance on livestock species for sustenance and food source in many parts of the world. The human coronaviruses, what makes things even more complex is when we look across the different coronaviruses. Certainly we knew prior to the emergence of SARS coronavirus and obviously the subsequent identification of MERS coronavirus and now SARS-CoV-2, coronaviruses were not that interesting from a human perspective because they were largely, associated with mild self-limiting diseases. And that really, I think took away our interest in vaccine development until we saw SARS-CoV emerge in the early 2000s. What makes things difficult for us is when we look across the host receptors that are allowing for attachment of these diverse viruses to our own cells, what we see is that there is not necessarily a common receptor that is used across all human coronaviruses. That now makes things a little bit more difficult. When we think about things like pan-coronavirus vaccines, we have to appreciate that while the spike protein is a common moiety that's held across all the coronaviruses, that spike protein is going to be highly divergent based on the receptor that it's going to bind to. So when we think about common epitopes and this idea of pan-coronavirus vaccines, it suddenly becomes much more difficult to try and identify those commonalities across the coronaviruses. And of course, then we have to appreciate that the host receptors that are involved in the attachment of those viruses onto our cells are going to be very diverse in terms of their expression patterns across different tissues and even across different animals. So this is not certainly an easy undertaking. Now, coronavirus distribution, unfortunately, is extremely complex, and I think we're only scratching the surface of our understanding of what this looks like. What we could say is that previously, when we look at the human or the viruses that are associated with human infections for coronaviruses, what we see currently is that the majority of these appear to have originated in bats. And we do see, though, that there are some human coronaviruses that do appear to have a rodent origin or a rodent reservoir. Where this becomes difficult is when we think about the vast diversity of coronaviruses that continue to be identified right now in nature. And certainly because of things like deep sequencing and deep genome sequencing, what we have to appreciate is our understanding is, and certainly our identification of different coronaviruses is growing rapidly. But the issue is that we don't necessarily know whether or not that equates to infectious virus when we make those identifications, and what the ramifications are for the presence of these different viruses across these species. So things continue to be very complex, and certainly when we think about mitigation of coronavirus spillover to humans, this becomes a much larger undertaking. And the reason that I say that is when we think about the reservoir host of a virus, certainly some of the viruses I work on, things like Ebola virus, we think the likelihood or the greatest likelihood is that bats are the reservoir species for the Ebola viruses. Monkeypox right now, don't really know. So we're in a position of trying to say, okay, well, how do we curb these spillover events and transmission events? Well, coronaviruses, for SARS and for MERS and likely for SARS-CoV-2, the likely reservoir host is bats. The problem is that this is not just as easy as being able to identify which bat species are infected by these coronaviruses and essentially trying to stop spillover. That's important for us, but it already is trying to identify a needle in a very, very large haystack, considering the diversity of bat species we see on the planet. The coronas have added an additional problem for us in that they also have the potential to be able to spill over to other incidental hosts. Certainly in 2002, 2003, we saw that the ramifications of those spillover events when we saw SARS coronavirus move into animals that were being traded in live animal markets, in particular animals like palm civets or raccoon dogs. And what we saw was basically the amplification of the virus within those species. And while the ultimate zoonotic transmission or the movement of that virus from those animals into human hosts was quite rare, what we saw was that when those events occurred and those patients were sick and moved into hospital settings, nosocomial transmission took over, and we saw the driving of the SARS coronavirus epidemic. In that position, though, we didn't see a lot of community transmission, which certainly allowed us to be able to try to get containment of that outbreak as quickly as possible. MERS, again, complicated story. At some point in the last 3 or 4 decades, it's theorized that MERS coronavirus moved from bats into camels, basically found essentially a new alternative reservoir host, has been circulating in these animals, causes mild illness when it's circulating within them, but can also spill back over into humans, and the route of transmission is not completely clear. Again, we see nosocomial transmission, but not a lot of community transmission. SARS-CoV-2 changed that perspective very quickly. Now, this is a preprint, controversial preprint by Mike Warby and colleagues looking at the emergence of SARS-CoV-2. The likelihood right now, at least from their data, suggests that at basically the Wuhan animal market, or the Hunan animal market within Wuhan in late 2019, there was likely an amplification event where we saw SARS-CoV-2 spillover from animals that were being hosted at that market into humans. Again, a lot of controversy surrounding this, but it should, again, galvanize our mind that this idea of bat-borne viruses moving into incidental hosts or intermediate hosts and then moving into humans is critical for us for being able to monitor further emerging viruses. For us, SARS-CoV-2, certainly from a One Health perspective, is not as simple, at least at this time point, as the story for what we see with SARS coronavirus, where we saw SARS-CoV basically move from bat species into a limited selection of incidental hosts, and then subsequent transmission to humans and circulation within humans. You can argue that some of this may be because the tools that we had available at the time didn't necessarily allow us to identify other animal hosts that may have been incidentally infected. When we look at SARS-CoV-2, this story has become very, very complicated. And what we've seen is certainly the reverse zoonotic transmission of this virus from infected humans into a variety of captive and companion animals and other wildlife-associated animals. What we've also seen in some of these species has been a continued circulation, and in some cases, indications of spill back into humans. Certainly hamsters, deer, mink, and now a single description of a cat being able to transmit this virus back into humans, again, brings up this idea that the complexity of this virus is going to continue to increase, not only with human-to-human transmission, but now this question of what is happening within animals. And where this is important for us is when we think about the spike protein, when we think about the protein that's sitting on the surface of SARS-CoV-2 and is mitigating the attachment to our cells and the cells that express ACE2 in other animal species. What we have to appreciate is that as these circulation patterns continue, whether it's within humans or it also includes non-human animal species, we start to see viral evolution. Now, whether or not that viral evolution drives the creation of a virus that is more transmissible, more virulent, or there's no change, we can't predict that. But we have to appreciate that evolution will ultimately change the virus within these species. So, certainly our interest and the interest of others is trying to understand what the mutations will look like within different animal species, what the really the susceptibility of different animal species is to different variants of concern, because we have to appreciate the virus has changed over the past 2 years. And how do we then bridge in our ability to do surveillance, not only in humans to look for new variants, but also in animals? And then how do we combat that from a therapeutic standpoint? So in regards to mRNA vaccines, and kind of going back to where this talk was originally going to go to, listen, mRNA vaccines have been in development for, if we look now at the original identification of mRNA, you can really say that vaccine development has been occurring over the past 3 decades, plus certainly since the 1990s onwards. There's been a really kind of concerted movement to look at the use of mRNAs either as a therapeutic or as a potential vaccine. And this graph and this timeline only go to 2014. That doesn't include what we've seen certainly through 2014 on, and certainly of the last 2 years. But the therapeutic applications are really, kind of, I think, ever increasing. So looking at these not only from the auspice of vaccines, but also for the delivery of therapeutics. And I think that's quite important, because when we think about mRNAs, we tend to just think about what these will look like from a vaccine landscape, but we have to appreciate that in the background, there are a variety of other infectious disease and non-infectious disease applications that are being investigated. So for clinical trials that have been taking place and are currently underway for mRNAs, what we can see is that the diversity of the infectious diseases and certainly the pathogens that are being investigated continue to increase. So whether we are talking specifically about RSV, influenza, HIV, chikungunya, rabies, Zika, I think what you're going to see is not only the already increasing landscape that we saw prior to COVID-19, but now an even more concerted effort to see whether or not mRNA vaccination can provide us with a scalable, rapid response to other emerging infectious diseases. Now, one of the things I do want to say, and I highlighted this today certainly in talking about monkeypox and what we're currently viewing as far as the global outbreak of monkeypox, I think we tend to not think about the global burden of emerging infectious diseases until they become a more global problem. So even though they've been circulating for years in vulnerable regions of the world, we tend not to increase our preparedness and response efforts until we actually see them move out of those regions. The problem is we are well behind the 8 ball by the time that those events occur, whether we think about Ebola, whether we think about monkeypox, whether we think about coronaviruses. We need to do a better job in terms of trying to get more preparedness and response, certainly infrastructure and investment in the regions of the world that are global hotspots, because those certainly are known to us. So when we think about mRNA vaccine design, these tend to work around basically a single-stranded mRNA molecule running in the 5' to 3' direction. This includes basically some additional sequences like the 5' cap, the 5' upstream untranslated region, the 3' untranslated region, and the poly A tail. All of these are meant to hopefully increase the recognition by host cell machinery, including the ribosomes, but also increase the half-life of these molecules. And of course, the central region of this is the most important, which is the open reading frame, which contains our antigen. One of the things that mRNAs allow for, though, is the addition of multiple antigens to a particular vaccine. So what we can think about is the fact that we may be able to create vaccines that cover say a broad range of variants that have emerged or are able to tackle multiple infectious diseases within one vaccine. The whole design strategy itself is, I don't want to say it's simple, But certainly it takes away from the need for the large kind of cell-based or even egg-based approaches that have been undertaken in the past. Basically, as we saw in 2020, once a sequence is identified, that sequence can be designed. Basically, that sequence can be put into plasmid DNA, and they can undergo in vitro transcription in large bioreactors. This allows for the large-scale production of the mRNAs that are presented here. And then through purification protocols, we can get highly purified mRNA, which can then be encapsulated in different lipid constructs, which we know are going to allow for the transfer of that mRNA into different cells or different antigen presenting cells following vaccination. So all of this allows for us to create highly specific mRNAs that also are able to target very specific cells. In our case, when we look at antigen presenting cells, these lipid nanoparticle-based mRNAs allow for basically the uptake of this negative charged mRNA into the cytoplasm of the cell. That mRNA is going to be released. It's identified by host ribosomes. The protein is produced, or whatever antigen we have provided the sequence for, and that is going to be exposed on the surface, as well as broken up into proteolytic byproducts, which will be recognized by cytotoxic T cells and will ultimately allow for not only the generation of a strong, hopefully cell-mediated immune response, but also a longer-lasting antibody response. And when we look at the efficacy of mRNA vaccines, we certainly have known through the past, certainly 2020, how efficacious these vaccines have been for COVID-19. Omicron has thrown an unbelievable right hook at us. We are still not at a place where we can predict what variant is going to come next. Omicron has pushed us purely for the fact it has been so unbelievably fit in terms of its ability to circumvent host immune responses and as well be able to transmit across individuals very, very quickly. Now, what we've seen is that certainly our protection from symptomatic infection with the current vaccines, the 2-dose regimens, was not very good. We still had good maintenance of vaccine effectiveness against hospitalization and severe outcomes. And we know that with boosters, whether they were through Pfizer or the Moderna mRNA vaccines, that we were able to see an increase in that protection from symptomatic infection. It's not long lasting, but it gives us some additional benefit in being able to try to control transmission to some extent within the population and bump up that protection for the most severe outcomes, in particular for those that are in those highest risk groups for severe infection. When we look at 3 doses of the Moderna vaccine against either the Omicron or Delta variants, we can see certainly with Omicron here in red that the days fall, or in the time since the second vaccination, that we saw almost a complete ablation of protection from symptomatic infection, but with a third dosage, that increases to at least to some level, not maintained for a long period of time, but again provides us with some additional benefit against transmission in the community. One of the things that's happened today is certainly the FDA advisory committee had met to talk about the pediatric vaccine for 6-month to 5-year age groups for both Moderna and Pfizer. They voted unanimously to recommend that that FDA does grant those emergency use authorization, and it's mostly been based on the data presented here, which really for a 2-dose Moderna vaccine showed that within that age group, about a 50% protective effect against symptomatic infection, and importantly, there was good immunogenicity that was found within that vaccine regimen as compared to other age groups. So that's pretty important. We're seeing good immunogenicity. The likelihood these vaccines are going to work. Pfizer at 2 doses did not see much of an effect, but with a 3rd dose, vaccine effectiveness rose up to 80.3% against symptomatic infection, but let's appreciate this was a small group of infected individuals with only 10 infection cases within the study group. So we need to be somewhat cognizant of those limitations, but again, there is good immunogenicity. So for people like myself who have a young child at home, I think we're hopefully moving forward. Now, in terms of more specific vaccines, again, one of the benefits of mRNA vaccines is you can adjust very, very rapidly. And we've heard talk about this since the emergence of Alpha through Delta and now to Omicron of when are we going to see variant-specific vaccines being pushed out. For us, I think we're moving very close to that. So Kyle Brown had sent me some of this data the last few days from Moderna that they had released in a press release, looking at their vaccine in combination with an Omicron spike vaccine candidate. So this is their mRNA 1273 vaccine with the Omicron spike, and that's now 1273.214. So with this booster dose for this particular vaccine candidate, what they saw was that they had good, really superior neutralizing antibody responses at a month post administration as compared to the original mRNA vaccine. And their binding antibody titers also appear to be significantly higher against all the VOCs as compared to that original mRNA vaccine. So it's moving in the right direction. We've got to appreciate, though, the virus continues to evolve and continues to change. So I think this is why you're seeing the movement towards pan-specific vaccines. Certainly for influenza the past few years, Florian Kramer and other groups have been doing this work, looking at trying to identify common epitopes across influenza viruses to be able to create a pan-influenza virus vaccine. So looking at the stalk region on the hemagglutinin protein, it looks like we're moving in that direction. We actually see vaccines that seem to give much broader protection against different flu viruses than what we've seen previously. That has also been adopted for these sarbecoviruses, and certainly I think there's more hope to be able to move this even more pan-specifically across human coronaviruses. Right now, a lot of this is trying to identify those common epitopes. Certainly Omicron, again, changed the landscape because certainly the number of mutations that we saw has drastically increased. So our, I think regionality for a common spike, even amongst SARS-CoV-2, is becoming smaller as this virus continues to evolve. But certainly with artificial intelligence and machine learning, the hope is that not only from looking at sequences, but also looking at those secondary and tertiary structures that are predicted, that we may be able to find common epitopes. And certainly in my field, moving beyond this to things like phelos, where we've identified recently Luho virus in bats in Hungary. We don't know what the potential for infection in humans is, or if there's any disease, but we have to appreciate that this is an important area. For emerging infectious diseases continue to evolve and change very, very rapidly. So with that, not presenting any real data from my lab, but just want to provide back some of the resources that we are using to try and combat this, whether it be through the Consortium for Emerging Infectious Diseases, through WHO, like we're doing with monkeypox right now, or through COVARNet here in Canada. I think what we're seeing is a very global response to a continuing issue with emerging infectious diseases. And I'll stop it there.
Excellent. Thanks so much, Jason, for a really fantastic and very comprehensive presentation. That was wonderful.
Thank you.
I'd like to encourage all of our participants to please type your questions into the Q&A. I think we've got time for a couple of questions, maybe one or two quick questions if we can. And perhaps I'll start with one, Jason, if I may, while I'm waiting for audience members to jump in. So I think you really nicely illustrated the complexities around viral emergence and transmission dynamics. And And you touched on just briefly about some of the different forms of surveillance, including animal surveillance. And I'm wondering if you could speak to some of the big lessons that we've learned around surveillance as it relates to both animal as well as environmental. I think I've been following wastewater surveillance for the first time. I think lots of us are learning about all the different ways that we need to try to watch for and better understand. So if you could share some of your thoughts, that'd be great.
Yeah, I'll give a couple of my thoughts. Listen, I think one of the things that we need to appreciate when we think about surveillance, and certainly when we look at I think the disparity in vaccine availability and uptake across the globe, what we have to appreciate is, again, most vulnerable regions of the world tend to be overlapped. Where we don't have good surveillance because there's not a lot of investment and not a lot of infrastructure. So in the case of SARS-CoV-2, one of the things that I would certainly implore the international community to consider is that we need to have better surveillance systems in the areas where we know we have low vaccine or vaccine equity issues, purely for the fact we need to be able to identify variants as quickly as they emerge. If we think about how quickly Omicron moved across the globe, we don't have months to be able to be able to try and meet these needs. We need to be ahead of the game in that regard. And certainly, again, when we think about beyond SARS-CoV-2, what we have to appreciate is that emerging infectious diseases continue to circulate. Monkeypox, great example, been circulating for years. It's endemic in many areas of West and Central Africa, but it became forgotten. And you've seen certainly the last 3 decades, people continually saying, listen, immune waning from smallpox vaccination programs, loss of people that received vaccination in the first place is going to lead to the potential for more global emergence. We're now facing that. The question will be whether or not we see increased investment in surveillance within those areas. Certainly in regards to the change, is there any change in transmission with this virus, or have we just not had surveillance to be able to tell us what is going on? So those things are critical. Certainly, again, we need to appreciate that investment not only has to happen at the regional, local, and national levels in surveillance strategies well beyond COVID-19, but also has to happen in certainly within the most vulnerable areas of the world.
Great, thank you. Another question that's come in is in regards to updating antigen and being able to tweak the mRNA vaccines, particularly for new variants as they come. So the question is, won't this just drive evolution of the COVID virus and prolong the pandemic?
Yeah, so this has been a great question, right? What is the driving force behind the evolution we're seeing in the virus? So all the data we have continues to say, listen, the likelihood for where many of these variants are coming from probably is in chronically infected individuals, unfortunately, right? And of course, when we think about, again, the disparity that we're seeing in vaccine uptake, well, we start to be able to see the picture of, okay, it's probably going to become more and more localized in areas where we haven't had a good vaccine coverage. So that tends to be one of our issues. The other issue is, again, the evolution in animals. We don't fully understand. That could be a potential driving force because we know that if we have unchecked evolution or unchecked transmission, the virus is just going to change. As we move forward, certainly, I think we are still in a position globally where we have a lot of people that frankly have not been vaccinated. And of course, immune waning as well has to be taken into account. So the evolutionary push on the virus to change based on immunity, I actually don't think we're at that point. There are still a lot of cracks in the system for this virus to be able to have ample time to evolve without having to respond to vaccination. But we have to consider that our best strategy is to get the entire globe vaccinated as quickly as possible and try and get transmission suppressed. If we can get transmission suppressed, we do reduce the risk of more evolution of this virus, because frankly, at this point in time, we can't predict what the virus is going to do next. Omicron is a great example of that.
Great, and an important message to close on. So with that, Jason, well, thank you very much for your really thoughtful presentation. And I'd like to move now to our third speaker and welcome Dr. Wasserman to the floor. Dr. Wasserman, welcome. Thank you.
Thank you very much. I'll just, okay. Anyway, thank you very much for inviting me. I'm delighted to be here. This is going to be a bit of a change of pace from a lot of the mechanistic and very important underlying basic science of this virus. So I'm an allergist, clinical immunologist, and I'm going to be talking today about COVID-19 vaccine-associated allergic reactions. These are my disclosures, which will not affect the content of this presentation. Anyway, I'm going to do a brief review of the data on allergic reactions to the currently available COVID-19 vaccines, and these will be largely focused on mRNA, which have been the most common culprits in addition to the ones that we have the most information about. Going to mention some possible mechanisms of anaphylaxis with the Pfizer and Moderna vaccines, and then just review potential strategies for safe re-administration of vaccine in those with previous reactions. Anyway, within 24 hours of mass vaccination, there were 2 reports of anaphylaxis in the UK and 6 in the USA. And the UK's Medicine and Healthcare Products Regulatory Agency, known as the MHRA, warned those with significant allergies, namely to vaccines and medicines and foods, not to get Pfizer-BioNTech's COVID-19 vaccine. And then just as quickly, there was damage control. This is just a quote from Dr. Paul Turner, who serves as in an advisory capacity to the MHRA and works at the Imperial College. We're tweaking advice to make it very clear that if you've got a food allergy, you're not more at risk. Clearly, the phones were ringing off the hook very quickly when this mass vaccination started and these initial trends were seen. And then in Hamilton, where I work at McMaster, we had our own bit of excitement. A Hamilton personal support worker says allergic reaction to COVID-19 vaccine led to fainting, seizures, and she needed CPR. Most of us who had the opportunity just to read the newspaper reports on this case concluded that this was highly unlikely to be an allergic reaction and more likely to be a vasovagal reaction, but she clearly got her few minutes of fame. So one has to be very careful when defining and making observations about vaccine-related anaphylaxis. There clearly is a differential diagnosis. Was there another cause at the time of the vaccine? Urticaria, angioedema, reactogenicity, very common with any vaccine or immune stimulant, and this isn't always consistent with anaphylaxis. Certain mast cell disorders like mastocytosis more often are able to give reactions at the time of vaccination. Flushing disorders, neurologic syndromes, and we've seen so many complaints like this, everything from brain fog to numbness to tingling, and this is not anaphylaxis. Vocal cord dysfunction, cardiovascular disorders, and a huge number of people really affected by stress, where sometimes it's just very difficult to tell the difference between an acute allergic reaction and things that may be seen with a stress disorder. But of course, we want our vaccines to be safe. And what do we know about anaphylaxis related to immunization? Anaphylaxis is an adverse event following immunization is rare. In the past, the numbers that have been quoted are something like 1 to 1.5 per million doses for most vaccines. This is a CDC number. And anaphylaxis associated with the Pfizer SARS-CoV-2 mRNA vaccine appears to be almost 10 times as high, anywhere from 5 to 8 per million dosages for Pfizer. And then as time went on, the CDC sort of refined that to 2 to 5 per million. These would have been the numbers circulated at the end of last year. Now, for anaphylaxis, most of them are IgE-mediated reactions, but they're also almost all to proteins when they do involve IgE, and they do require prior exposure. And what threw us all for a loop is that these vaccines, which are mRNA, do not contain proteins, and reactions were reported after the first dose. This is from a recent systematic review, because by this point in time, we've had opportunity to really review the millions of patients and reactions which they've had. This was just published recently in the Canadian Journal, and the numbers are not much different than what's been quoted sometime last year, namely 8 cases of anaphylaxis with the Pfizer vaccine per million doses administered, and for the Moderna less at about 2.8 per million. So what did we do when we first found out about all this? And really the first thing we did was reach for the vaccine ingredients. What was in there that could possibly be triggering this reaction? For both the Pfizer and the Moderna, one of the things of note, since both are chemical soups of all sorts of things not generally associated with allergy, was polyethylene glycol. Both of the vaccines contain PEG at a molecular weight of about 2,000. Another component that was found in the Moderna is something called tromethamine, and this is found in a number of radio contrast dyes and has very rarely also been associated with anaphylaxis. For the AstraZeneca and the J&J vaccine, the excipient here is polysorbate 80, and I should note that both polysorbate 80 and PEG are found in a huge variety of products, which people will encounter every day just in the course of using household products and medication. So the initial attention was focused on those, at least for PEG, because the mRNA vaccines appeared to be causing most of the trouble. Now, in the allergy world, we've been very familiar with certain things causing allergic reactions like egg protein, formaldehyde, gelatin, and MMR, neomycin, thiamerosal, but PEG and polysorbate were entirely different to us as being vaccine components. They are excipients. They're used to stimulate the immune response, to stabilize the vaccine, and to prevent contamination. And there are 4 possible mechanisms where these vaccines could possibly induce allergic reactions. Specific IgE production by activating antigen-presenting cells and Th2 cells. Complement activation products directly can be activated by the vaccine, which can activate mast cells, direct mast cell activation, or by stimulation of the inflammasome. What was unusual is that in the large clinical trials done with both of these vaccines, the Pfizer and the Moderna, there was very little anaphylaxis seen. In fact, the vaccine group prevalence of anaphylaxis was the same as the placebo group. But to be fair, both Pfizer-BioNTech and the Moderna vaccine trials excluded those with any severe allergy to any component of the vaccine. So they were already quite pre-selected. And the Pfizer vaccine specifically also excluded participants with a history of severe allergy to any vaccine. So throughout the clinical trials, there was probably 1 case of anaphylaxis with the Pfizer-BioNTech and none for the Moderna. So what did the initial cases of anaphylaxis look like? And this was from a publication early in 2021 in JAMA, where they characterized 66 patients who had had anaphylaxis after an mRNA vaccine. 32% of these patients had had prior anaphylaxis. All of them required treatment in a healthcare setting. 48% were hospitalized, including 18 in the ICU, and 7 were also intubated. So not a small thing initially. And what were our resources at the time when we were starting to contend with this? Because these sorts of issues were taking over all the allergy clinics very rapidly. What we had to work with was a task force report from the American Academy and the American College of Allergy and Clinical Immunology outlining adverse reactions to vaccines, but this was last published in 2012 and had nothing to do with the current mRNA vaccines. We also had our experience with vaccine anxiety in egg allergic individuals when we were giving in 2009, the H1N1 vaccine at the time of that. We also had our own knowledge. We also had a guidance written by the Canadian Society. And we also had a paper which was published early in 2021 with an expert group from the American Academy who outlined an approach to this. But at the end of the day, just to give you, the sort of background that we had very little to deal with in trying to figure this out and how to approach vaccination in large numbers of patients who were allergic. These were the initial recommendations that came out from the Canadian Society of Allergy as well as NCCID. And the Americans had very similar recommendations as well. Anybody who required vaccination, who had any history of significant allergy, be it to foods or drugs or venoms, etc., both organizations recommended that these people could be vaccinated without any special precautions with just 15 minutes of observation. For those who had a severe allergic reaction to any injectable therapy or vaccine not related to an mRNA vaccine, NCCID recommended a longer interval of observation, namely 30 minutes. And for those who had a history of a severe allergic reaction to a previous dose of the vaccine, or suspected or confirmed allergy to any of the components, what both organizations recommended is see an allergist. The Canadian Society said considered graded vaccine administration for future doses, but none of us had any experience breaking up an mRNA vaccine into a graded dosing schedule. They also said that allergy testing to the excipients like PEG was not recommended because we really couldn't comment on the feasibility or validity, both of which were unknown, and that pre-medication was not really necessary. NCCID had sort of a similar view. In general, if somebody had a severe reaction to a first dose, what they said is the vaccine should generally not be offered, but if the benefit outweighs the potential risk and informed consent is obtained, a COVID-19 vaccine using a different platform may be considered. So most of us who were seeing these patients in droves at the beginning, complaining of different types of reactions, found a lot of this very unhelpful at the time, especially since we did not have any vaccine to administer within our own clinics. And we were sending back people to vaccine centers and pharmacies to receive dosing with really no guidance. So what have we learned? One thing that we learned is that we're able to immunize highly allergic individuals. The fear at the beginning did not materialize overall over the course of this experience. This is from one study published in JAMA in 2021. They took about 430 patients, all of which had a history of allergy. They gave them their first dose of an mRNA vaccine under medical supervision. 98% had no allergic reaction. A little over 1% had a mild allergic reaction, and under 1% had anaphylaxis, so a low rate of about 0.7%. When it came to the second dose, and 218 of these patients received that under medical supervision, again, 98% had no allergic reactions, and close to 2% had minor allergic reactions. So the second dose appeared to be even less problematic than the first, and everybody at least was able to receive a first dose and half the second. So let's move into some of the excipients and, people who may have a problem with polyethylene glycol, since this was implicated as a potential problem when all of this came to pass. We've mentioned that PEG and polysorbate improve water solubility in drugs and vaccines. PEG is used to stabilize the lipid nanoparticle containing the mRNA. The PEG in these vaccines is 2,000 molecular weight, and it's larger and of different sizes in many healthcare products. It's a prominent component in bowel preps, ultrasound gels, dermal fillers, cosmetics tablets, etc., but of a different molecular weight than what you see in the vaccinations. One of the studies looked at whether or not testing to PEG specifically was useful to predict COVID vaccine allergy. They looked at 65 patients with immediate allergic reactions to the mRNA vaccines. Only 4 of them had a PEG positive test. 2 tolerated a second dose, 1 tolerated a Janssen vaccine, and 1 opted not to get vaccinated. But of the 65 patients who received a second dose, 75% had no reaction, and the rest were mostly cutaneous, but 2 did need epinephrine treatment. But the overall conclusion of this paper was excipient testing did not add value to the risk assessment, and most individuals with an allergic reaction to the first dose of the mRNA vaccine could receive the second safely, regardless of skin test results. And yet, PEG has now arisen, at least in some case reports, as a cause of anaphylaxis to the Pfizer-BioNTech. This is just one early report of a 52-year-old female after the Pfizer vaccine who developed very typical symptoms of anaphylaxis. She required treatment with epinephrine, cortisone, and fluids, and she'd had a history of anaphylaxis to azithromycin, to shampoos and shower gels, which in retrospect were PEG containing, but of a different molecular weight. This lady was skin tested to both polyethylene glycol, polysorbate, and she was tested to a variety of different molecular weights of PEG, which was done in this study. When she was skin tested to the PEG 4000, she developed a severe systemic reaction after 12 minutes, and the PEG was felt to be causative in this particular vaccine. And then shortly after, there were 2 other published cases with Moderna, also proven to be PEG. So there's always the individual where testing may be beneficial and where it is a proven cause. What's come up since then as well is that polyethylene glycol, when somebody is given that allergy label, is not an absolute contraindication to receiving an mRNA vaccine. This is from a retrospective chart review of patients who were listed as having PEG allergy amongst their allergies, and they presented to a COVID-19 vaccination clinic. Now, how these allergies were substantiated, many of these people had reported initial gastrointestinal intolerance or cutaneous symptoms on PEG exposure or other allergic symptoms. And they did have data on 100 of these patients listed as being PEG allergic who underwent vaccination. And all patients tolerated the full vaccine series with no allergic reactions in this particular series. So having this as a label was not a contraindication at the end of this study, in that most people could be safely vaccinated. So let's move on to those people who've experienced an allergic reaction to the first dose. Can they safely receive a second mRNA COVID-19 vaccine? Again, this is a study that examined the safety of a second dose of Pfizer or Moderna in those with allergic reactions to the first dose. This was a group of 189 patients, 17% of whom had had anaphylaxis to the first dose. And you see the most prominent symptoms that they complained about. Of these 189, 159 of them received a second dose, 47 of the 159 were pre-medicated, and 32 of the 159 did report some immediate allergic symptoms associated with the second dose, but these were felt to be mild and resolved easily with antihistamines without requiring epinephrine. So this is also somewhat unusual. What about testing with the COVID-19 vaccine itself, since we test with everything in allergy? And there were a number of barriers to being able to do that. Number one, we had no vaccine initially. The vaccine supply was limited. It's a fragile vaccine with temperature controls, time limits once it was thawed and diluted. So we really had very little to work with. And it was also a new vaccine. We had no idea of the sensitivity or specificity once we did skin test with it, nor the minimal inhibitory concentration. So this never made it into guidelines as being a useful strategy to predict risk. Along with anaphylaxis, there are lots of other significant adverse effects that people complained about in our own clinic as well, not only anaphylaxis, but vasovagal, sympathetic stress reactions. And looking at all the different symptoms and things which characterize these individual afflictions, they're all fairly similar and sometimes hard to tell the difference. which is why when a lot of these cases were gone through very thoroughly, not all of them did meet a definition of anaphylaxis. And it was felt that many of these could have been secondary to stress or vasovagal responses. And of course, the nocebo effect. In drug studies and vaccine studies and in real life, many people will respond either to a placebo effect up to 32 to 35% in some of these trials of vaccines. So that's not to be forgotten either. So let's just look at a number of cases and what we've done in our clinic. This is a first case of a COVID arm, which is a delayed inflammatory reaction, extremely common, primarily seen with the Moderna vaccines initially. This is a case of an 86-year-old female who had a delayed reaction a week after administration, swelling and redness at the site of injection. She was treated with antihistamine. And on her second dose, she was pre-treated with an antihistamine called rupatadine night before and day of, and then received her second dose 3 days later, had a bit of swelling, felt to be a lot less severe, and again, the dose was tolerated. We've seen a lot of cutaneous reactions of all types, delayed erythromelalgia, lots of urticaria, which have been described, local site reactions. The messaging is they start within the first week. Many of these can start as late as 7 or 8 weeks, and some of these cutaneous reactions can go on for a long time. So be it natural infection or be it vaccine related, lots of these adverse events have arisen and they're hard to prove, but everybody gets revaccinated when I see this. This is another case, 34-year-old nurse, past history of allergic rhinitis and asthma, who received her first dose of Pfizer, developed hives on her upper chest, itching at the injection, facial numbness. She was admitted to the ER for observation, had stable vitals, and treated in the usual fashion with Benadryl 25 milligrams and prednisone 50. On coming into our clinic, we do skin test everybody. We do it to PEG, polysorbate, latex, chlorhexidine, which is often used to wash the arms of these patients. She was pre-medicated in advance of her second vaccine with rupatadine 20, famotidine 20 the night before and day of the vaccine. She received her second dose with 30 minutes of observation, and it was received without a problem. So in our clinic, and I'm certainly happy to defend this, we both skin test and we do pre-medicate, unlike the usual recommendations which are given by the Canadian Society of Allergy. This is another case of an allergic reaction, 50-year-old radiology technician, history of latex allergy and asthma, developed facial numbness, mouth itching, cough, lightheadedness, and erythema at the site of injection after her first dose. Again, Benadryl 25 milligrams and prednisone 50 in the ER. She was skin test negative to all these components when she came to see us before her second dose. She was pre-medicated as well, this time somewhat more aggressively with cetirizine, famotidine, montelukast, which is used in a lot of pre-medication protocols, and again, Pred 50 the night before and the day of. Now, this individual had a similar reaction as she did the first time, but it was a lot less severe, and she was able to go home at 30 minutes instead of presenting to the ER. This is somebody with a case of myocarditis, 69-year-old male who came to clinic with no previous cardiac history. He received the Pfizer vaccine and 2 weeks later was in ICU with myocarditis and in fact was quite sick and not of your typical age group to have had this complication. But he completely resolved and was discharged home with a normal echocardiogram and ECG. He wanted to be revaccinated for social and travel reasons, and we went through our usual protocol with a lot of anxiety and shared decision-making on my part. But he was vaccinated, and luckily we were able to get another platform with AstraZeneca. He received his second vaccine without difficulty, his third dose of Astra without difficulty, and has just had his fourth dose with Novavax, again with no recurrence. So these people with a different platform have been able to be safely revaccinated, but even with a repeat mRNA platform, we've had a couple of patients in our clinic who've insisted on it and with pre-medication had no recurrence. Again, these decisions are not undertaken lightly, but they're doable. Why do we pre-medicate? Because as allergists, this is what we've been doing for other similar substances like radio contrast media. This is another example of something which can lead to an anaphylactic reaction, where if it's given again, there's up to a 50% chance of a repeat reaction, and we have no available methodology to identify those who do react again. These are thought to be complement mediated. There doesn't seem to be any IgE involvement, but with pretreatment with antihistamines and corticosteroids, the rate of reactivity on the second exposure to radio contrast goes from 50% to 5%, and the repeat reactions are usually mild. So basically, we went with what we knew, even though there was no evidence. This is the US experience regarding allergy. 318 million Pfizer-BioNTech doses have been given and 21 million Moderna, and there have been no deaths due to anaphylaxis, and the same in Canada and worldwide. So these are my take-home messages. The incidence of immediate reactions to mRNA vaccines, they're rare, but they appear higher than to the prior vaccines, which are non-mRNA. Most of the immediate reactions are not IgE mediated, so you have to consider also alternative diagnoses, including stress responses. The risk factors for immediate reactions to mRNA vaccines are different from the prior vaccines. We're seeing them as being younger age, female sex, allergy history. This was what was noted in those initial 66 cases, and the messages seem to have been borne out in some of the later cases as well. Most reactions are first dose and do not recur with dose 2, also unusual for things associated with allergy. In our clinic, no allergy history is an absolute contraindication to revaccination, and the message is see an allergist. Kaiser data from the USA shows that about 21% of vaccine reactors did not get a second dose. No anaphylactic deaths have occurred across 460 million mRNA COVID-19 vaccine doses. And at our clinic, which is at the Firestone, St. Joseph's Healthcare Hamilton, we do skin test to the vaccine excipients, not because these are reliable predictive reagents, but I can tell you that the psychological benefit of seeing a negative test in people who would not otherwise get immunized is huge. We do pre-medicate, as I mentioned, and we vaccinate. We've been lucky within our institution not only to have access to the mRNA vaccines, but prior AstraZeneca and Janssen, the Janssen vaccine, and now Novavax. So every attempt is made within our clinic to safely vaccinate. So with that, I thank you for your attention and would be happy to address any questions.
Susan, thanks so very much. And no surprise to me, what a wonderful presentation. I think you've helped us mine through a real minefield, because I think as both infectious disease specialists and allergists, we've been dealing with these questions on a rather regular basis. So I always will encourage, obviously, folks to send in questions. You can send them through your Q&A, but while they're thinking about it, I think in the interest of time, we do have, I have a question I had that I was wondering if you could field. Sure. So I think, Susan, you've made it fairly clear, and I think it's been very helpful for us to understand that the majority of reactions are not IgE mediated. And in fact, the majority of reactions are probably not true anaphylaxis. But we still get these requests. I had one just a few weeks ago about people still seeking these exemption letters. Is there any phenotype or group of patient that you would define that you think should or could qualify for an exemption letter?
Look, Sharik, I've been seeing a lot of these sorts of patients. Everybody wants exemptions. Look, if somebody has had a severe allergic reaction to the first dose of vaccine or myocarditis, these are the accepted and recognized indications for getting an exemption. So if somebody did not want to go through my protocol of being pre-medicated, vaccinated under supervision, if they had undergone either of these, then I would exempt them if they were unwilling. I have not exempted many people. The other who I did had a Miller-Fisher syndrome, the chronic Guillain-Barré variant, was vaccinated, had to be admitted to hospital for intravenous gamma globulin. That individual, a woman in her mid-70s, I understood that getting revaccinated, even though I offered her a different platform, she didn't want to go through that again, and I thought that that was legitimate. The number of patients who I have seen, and I'm no stranger to things like brain fog. I mean, we see all of this mast cell related disease and whatever, but people who complain of, year-long poor concentration, numbness, tingling, unwellness, fatigue, looking to be exempted. I just don't. I can't. I have no way of showing any association with the vaccine. It's not a recognized exemption, and beyond trying to do my due diligence and rule out other causes, I can't go beyond that.
Thanks very much, Susan. On a personal anecdote, thanks for helping my mom out, because she had this issue of severe allergic reactions, and through your pre-medication protocol, she flied through her third dose and fourth dose, in fact, of her vaccine boosters, ended just fine. So thank you for that very pragmatic
Anytime,
overview.
Sharik. You know what, look, I think that what made this very different is that at the time that we were told, don't test and don't pre-medicate, we were turning people back out to the community to just be vaccinated. Not only was the community not willing to do it, and we had plenty of people sent back saying, well, this person had a reaction, we're not vaccinating, but a lot of people didn't even want to go. They lost trust in us, basically. So, I think it's important that we offer something, even if we don't understand the problem totally.
Thanks very much, Susan. Well, listen, everybody, in the interest of time, this brings us to the end of our webinar, but I wanted to really end this first by thanking a number of people. I should obviously start by thanking our two co-moderators first, Dr. Cooper and Dr. Becker, for supporting this evening's talk. And I have to thank our three speakers. And I was just thinking that yesterday there was an update sent by the World Health Organization telling us that, we had reduced the peak of infections by 90% and the peak of mortality by 90%. And clearly the vaccines played a major role. So today's vaccine webinar is a timely reminder of how important this intervention was in the pandemic. And I want to thank Dr. McGinty for that wonderful overview of both current vaccines and the future of what's still coming down the pipeline. Dr. Kindrachuk, obviously, for walking us through the evolution of coronaviruses and trying to understand the spillover effect that we're seeing and the need for ongoing surveillance and the importance of access and distribution. And obviously would like to thank Dr. Wasserman for that very pragmatic approach on how to identify, deal with, and really separate, the truth from myth, which I think has been one of the biggest challenges as we've been dealing with these folks. I think it's very important tonight also that we thank our sponsors for the night, Moderna Pharmaceuticals and NCCID. Without their, obviously, contributions, this would not be able to be pulled off tonight. And I wanted to end on a humbling note that I thought it was really heartwarming for me to see Mickey's cat and Jason's little one behind the scenes, because it reminded me about the fact that though we have all these amazing professionals in the field, they've got these personal lives, and the ability to balance that personal and professional lives is incredible. And it was really heartwarming to see that today. So thank you all. I obviously want to thank our attendees for sticking along with us for this hour-and-a-half webinar. And again, I remind you to visit the CFID website. There are opportunities, obviously, for you to both get engaged with CFID, and certainly if you're interested in supporting our CFID activities, that would be most appreciated. So on that note, on behalf of Dr. Cooper, Dr. Becker, and the rest of our CFID Board of Directors, I'd like to wish you all a wonderful evening, and thanks very much for attending. Have a good evening.